Welcome to the last section, or the last video on the second section, where we're talking about amino acids and protein structure. And now what we're going to do is we're going to start talking about if we have a favorite protein, how do we go about to study it? And so we're going to talk about the different ways, the different techniques we can use to study our protein. And that's because proteins have to be purified so they can be studied. Because when we're talking about proteins in the context of a cell, we're talking about hundreds, if not thousands hundreds and thousands of proteins that are going to be in there. And so we can't just get a cell, get a protein, and just study it. We're going to have to purify it so that we can get it alone from all the other stuff. So the first step, and what we're going to do is we're going to take a liver. Let's say our protein we look at is in the liver. And what we have is, we have a blender, and we have a buffer. Right? And so what we do is we put the liver into our beaker with a buffer, and then we just blend it up. And what we get is what's known as a crude extract. Oops. Let me go ahead. A crude extract. And so within this crude extract, we have a bunch of different proteins. And the proteins that we want is that one right there. This is the protein that we're looking for, so we need to ID it. So how are we going to go about doing this identification of this protein and then this subsequent analysis? So that's the first step. The first step is we get a crude extract. We get a bunch of proteins in a solution. The second step is going to be, we're going to then separate our protein of interest, this triangle with the red circle around it. We're going to try to get that protein separated from everything else. And there's three major ways that we do this. We can do it by charge. We can do it by size. And we can do it by what's known as affinity. And so what we have is charge is usually done through ion exchange chromatography. The size is usually through size exclusion, chromatography, or dialysis. And then the affinity is done by affinity chromatography. Forgot my eye. Right, it's through affinity chromatography. Now, one of the things to keep in mind as we talk about these is that we're talking about chromatography, right? And so in chromatography, there's always a solid phase and a mobile phase. And so one of the things we need to just keep in mind is what's the solid phase and what's the mobile phase. So we're actually going to look at 2 first, and then we'll go back to 1 and then 3. So let's look at size exclusion chromatography. So size exclusion chromatography separates proteins based on their size, based on their molecular weights. And so we have our column, and we have beads in the column. These beads have, I'll just draw some bigger so that we can see. These beads have pores in them. And so what happens is, these beads are known as Cephidex beads. And they come in, and the pores come in a variety of different sizes to separate a variety of different molecular weights from each other. And so what you have is, we have large proteins, medium-sized proteins, and then we have our protein of interest, which is pretty small. And so what happens is, the large proteins will not enter into those pores, and they'll elute first. Right? The larger proteins will elute first because they cannot enter into the pores. The medium size may occasionally hit 1 or 2, and then they'll come out second. And then the third the smallest protein will hit almost every single pore on the way down, and it will come out last. So, with size exclusion chromatography, big proteins, large proteins come out first, medium proteins come out second, small proteins come out last. This will be opposite of what we'll see when we talk about gels, when we talk about electrophoresis, because usually in electrophoresis it's smallest, medium, large. Here it's large, medium, small, and the reason is because, like a person at a playground, if a large adult were to try to go through the jungle gym, they will get trapped because they can't get through the thing fast enough. So that's why in electrophoresis the large ones are slowed down, whereas this one, instead of going through the jungle gym, the large protein just goes around it, whereas the little one still goes through it, or a smaller person can still go through it. And so large first, medium second, smallest last for size exclusion chromatography. Ion exchange chromatography does not separate proteins based on size, but based on charge. Right? Could be also called charge chromatography. Usually it's known as ion exchange chromatography. And so again, we have a column. And in this column we have usually a structural support of some sort of a bead. And in this case, we'll have a sulfate group on it, which will be highly negatively charged. And so what's going to happen is, to this column, let's go and draw two of these beads. To this column, we're going to do what's known as charging it. And what we're going to do is we'll do first, we're going to add calcium. And what that calcium is going to do, it's going to be coordinated, that ion, between those two oxygens. And so what's going to happen is, let's say we have three proteins. The first protein is our protein of interest. And we know our protein of interest is slightly positively charged on the outside. And then we have our second protein, which is just has a slight negative charge. And then we have our third protein, which is a square, which has a really highly negatively charged on the outside. So when we put this in, the first one that will elute will be the one that's positively charged, because you'll have that charge-charge repulsion. This is known as anion exchange. So the example that I'm using is an anion exchange column, in that it will bind to negatively charged proteins. So the positively charged protein will elute first. The slightly negatively charged protein will elute second. And the final one will elute last. And the big key, and the reason why isoelectric points are very important, is because of this type of ion exchange chromatography. pH buffers changing will allow these changes in charge of these proteins to allow for elution or binding of these specific proteins. And so that's the second type. Now we're going to go into the most powerful of all of them, but we also need the most knowledge of these proteins. And that's known as affinity chromatography. And so what happens is, what we can do is we can charge up another column. I'm just going to show one side of it. We can charge it up with copper. And let's say we have something in our protein that can really bind to copper. So it is going to bind to this. So let's say we put this is our protein, we put it in our column, and it's going to bind to copper, while all the other proteins just flow out. That's awesome. We have bound our protein. But the problem is we want to get our protein off so that we can study it. And so what we would need to do is outcompete this protein off of the copper. And so usually when you use copper, you use something like imidazole. Right, so there's imidazole. Will be thrown in there. It will displace the protein, and we'll get our original protein eluted out so that we can study it. So the big key and the big reason why we do size exclusion, ion exchange, or affinity chromatography is to study the protein's activity in its native form. These are non-denaturing conditions. We do not denature these proteins. So, what if we just wanted to know the size of our protein? We don't really care about whether it's natured or denatured, whether it still has its function or not. We can do what's known as SDSPAGE electrophoresis. So SDS is sodium dodecyl sulfate. This is what it looks like. Here's the dodecyl. Here's the sulfate, with the sodium as the counterion. So, SDS. So what we can see is if we were to split this molecule right here, that to the left is hydrophobic, to the right, hydrophilic. And so what we're going to do is this will get into the middle and the glob of the protein, and we'll start circularizing it even more, kind of making it more homogeneous, and it's going to give it a net negative charge. And so the net negative charge is good because we can calculate mobility of a protein. It's equal to its velocity divided by its electrical potential, or the net charge divided by the frictional coefficient. Now, we remove these two things when we add SDS because there's a net negative charge, and so we can just worry about separating these proteins really based on size. There is some shape problems or caveats that we get into when we talk about SDSPAGE, but we're not going to worry about those. And so what happens is it collapses from a native shape to a sphere, so we can just worry about separating them based on charge. Just to let you know, one SDS unit usually goes to 2 amino acid subunits. So that's SDS. PAGE stands for polyacrylamide gel electrophoresis. So let's look at acrylamide. Let's do it in a different color. I know this slide is pretty busy and I apologize, but we'll just put it all on the same one. Here's acrylamide. What happens is free acrylamide, unpolymerized, is very toxic. It reliably causes cancer. It's a neurotoxin. It's a very powdery substance. So usually we have to be very careful around it. When it becomes polyacrylamide, when it becomes a gel, it's very non-toxic. Just to let you guys know, if you like French fries or you heat carbohydrates at a high temperature, you do get some acrylamide. Not enough to be worried though. So let's say we have sample 1, sample 2, and we always run a ladder. So we have a ladder, positive side, negative side, because again, there's a total negative charge. And then what we can do is we can look at these proteins and we can say, based on the size of these proteins, they're different. They're different proteins in the sample or whatever you want to argue with. So the separation is based on size or molecular weight only. So now what we can do though is we can also put a gel where we have a pH gradient. Let's say this is pH 9 going to pH of 3, and we run it. So we put a protein on this end and we run it to the right towards pH of 3. This will be positive, this is negative, and what you see is you'll see a little dot, a little bit bigger dot, a large dot, and then a little bit, and then a small dot. And this right here is the pH that equals the PI, right? That'll be the isoelectric point. It's known as isoelectric focusing. It's a way that we can determine the isoelectric point of a protein. And what we can do is we can couple these two together and do what's known as a two-dimensional gel, where we look at both the isoelectric point and the size of these proteins. And so it's a very powerful tool, especially in things like cancer, and when people study cancer cells and a variety of different tissues, they use a lot of two-dimensional gel electrophoresis. So we're going to end this here. I'm going to not worry about talking about how proteins are sequenced. Edmond degradation, Sanger sequencing of proteins, you can look them up on the internet. The most common way we now sequence proteins is through mass spec. And so if you find that interesting, I would recommend looking at those and just learning more.